Cancer is the second leading cause of death in the world. In Canada, it is estimated that 1 in 2 Canadians will be diagnosed with cancer in their lifetime. 1 in 4 are expected to die. Despite its prevalence and the incredible medical breakthroughs on the forefront of cancer research, the number of deaths caused by cancer remains significant. Conventional therapies such as surgery, radiotherapy, and chemotherapy bring about a host of side effects with limited success, and they have not supported our long-term advancement in the fight against cancer. But there is a light at the end of this tunnel, something known as oncolytic viruses. Oncolytic viruses, as the name suggests, are naturally occurring or engineered viruses with a hunger for cancer cells. These viruses have a selective nature for replicating in and killing cancer cells while leaving properly functioning body cells intact. A little microbe that cures cancer without destroying our body in the process? Wow, oncolytic viruses sound like a magic cure. But how were their superpowers discovered? When oncolytic viruses were first used in the context of cancer intervention, the theory was that they would act as the active agent in the therapy to stunt tumor growth within the patient. Researchers first noticed and understood the applications of these little killers through a phenomenal observation of tumor growth impediment and regression after a viral infection in cancer patients. These early observations were not a result of an attempt of an alternative novel therapy, but rather were the result of an accidental contraction of infectious disease in these cancer patients. Interestingly, this actually resulted in clinical remission within these patients. From these crazy observations, therapeutic research on oncolytic viruses emerged. The most promising result in the battle against cancer using oncolytic viruses emerged through the use of adenoviruses. In 1956, these viruses were used to treat 30 women with advanced epidermoid carcinoma of the cervix. Within 10 days, in 2/3 of these women, tumor necrosis was observed, almost exclusively in cancerous tissue, indicating selectivity of the virus to replicate solely in cancerous cells, leading to minimal side effects. Revolutionary observations, right? This is why adenoviruses are the most widely used oncolytic viruses. Adenoviruses have a similar appearance to any common virus. Picture what they taught you back in grade 11 biology. An icosahedral-shaped capsid, so the big head, some capsid proteins sticking out of the head, notably hexon and penton fibers, and finally the viral, non-envelope, double-stranded DNA within this head. The question then becomes, why are adenoviruses such good cancer killers? And how do they get into cancer cells while leaving the normal functioning cells virtually unaffected? These are the questions that trumped scientists too. The truth behind the effectiveness of oncolytic viruses is due to the diminished viral infection protective mechanisms in cancerous cells. Normal cells have a protective mechanism in place against viral infection. However, in cancer cells, these mechanisms are impaired, allowing oncolytic viruses to enter and replicate effectively. An infection in a tumor cell is initiated when the adenovirus fiber attaches to receptors on the surface of tumor cells. From there, a second recognition occurs in which the virus penton fiber interacts with the tumor cell integrin receptors, allowing the virus to enter the cancerous cell. Like with other viruses, the host cell replicates the viral DNA, producing viral progeny and disruption of the cell membranes, and eventually death to the tumor cell. Viral particles and tumor antigens are released from the lysed tumor cell, promoting the immune response to kick in. At last, the immune system mitigates the viral infection, leading to remission. This all goes to show that in theory, these adenoviruses are very promising cancer killers. They can also be genetically engineered to recognize cancer cells using other mechanisms, such as in the case of low receptor expression on tumor cells. Such recognition mechanisms would increase the efficacy and decrease toxicity side effects of adenovirus therapy. These oncolytic viruses have the potential to completely revolutionize the way physicians treat future cancer. In theory, the specificity of oncolytic viruses makes them the perfect predators for destroying cancer from within, producing minimal side effects and toxicity in the process. Another oncolytic virus, Oncorin, was designed rationally for tumor selection and has been in use in China since 2005 as a therapeutic treatment for solid tumors. Oncolytic viruses are on the forefront of cancer research, and new therapeutic viruses are being developed every day with the ambition of treating bigger and badder cancer types. Unfortunately, in its current state, oncolytic viruses have not shown effectiveness in some types of late-stage cancers, like glioblastomas. Future aspirations of scientists in this field is to develop selective viruses for such cancer types. In terms of glioblastoma research, there are multiple early-stage oncolytic virus therapies in their testing stages, showing some promising but limited results. The emergence of oncolytic viruses provides a new potential strategy for us to alleviate or even cure malignant tumors. As research continues, its development will provide promising treatment options for numerous cancer patients.